ethical issues. Now, in May last year, this paper appeared in a journal which is not very well known, Protein Cell, CRISPR-mediated gene editing in human tripronuclear zygotes. Now this paper, as it turns out, was not accepted and was declined by Nature and Science. They didn't want to publish it. Tripronuclear zygotes are zygotes which are transformed by two semen. Normally, in vitro fertilization or in normal fertilization, there's only one male and one female nucleus get together, and the zygote closes itself for a second one. But I think about 3 in 100 cases, two cells get into the oocyte, and they are then called tripronuclear because obviously they have 3 nuclei. And they can be implanted into a uterus, but they will not develop in any way. Now, so that was, I think, in May. I remember very well, and this is the title. And actually, that experiment is quite disturbing in a way, but I come to that in a second. Because they used 74 human embryos to do this experiment. 74 human embryos which were tripronuclear. Since the yield is very low, as I said, 3% of the inseminated human embryos are only tripronuclear. They must have worked with a couple of hundred human embryos. Now you have to know, just to see the ethical dimensions, you have to know that in our species, Homo sapiens, superovulation only creates 3 or 4 embryos. Normally 1, but if you superovulate, it's 3 or 4, and in mice it's 100. So in mice it's routinely done in the laboratory. In our species, it doesn't work very well, and the cost of this for a woman is rather high, because you can imagine what it involves, and it's an effect like the climacterium can be, and then you get only 4, so you need hundreds of women to do this. And for me, I'm not Catholic or anything, but I think that I can understand that you do these kind of things for the purpose of having babies. I think the desire to have children is extremely strong, and that is justified. But to justify it for research purposes, this we have to discuss. I wonder what you think about it. In any case, even before this experiment was published, I think the group is working in Guangzhou at the Sun Yat-sen University. Sun Yat-sen was the founder of China just before World War I. Before that was published, people in the US had a meeting already on the initiative, I think, of Jennifer Doudna, a meeting in Napa Valley in January last year, about a year ago, a little bit more than a year ago, to discuss these implications. And why was that? Because there was a meeting in February 1975 in Asilomar, California, which is in Monterey, south of San Francisco, where the original recombinant DNA discussion was led. And you remember, no, you don't remember, but at that time the situation was rather critical. I mean, it was somebody called Paul Burke, who later got a Nobel Prize, a very famous biochemist, who wanted to clone pieces of a tumor virus, SV40, into bacteriophage lambda, which is a bacteriophage which under certain circumstances has a circular genome. And he wanted to transform and then this way kind of create lots of DNA in order to study it in more detail. Don't forget that was the time before DNA sequencing there was no way at the time how we could isolate genes, and it was a mess. We didn't have electrophoresis. We had sucrose gradients. We had laboratories filled with 20, 30 ultracentrifuges. This is all we did at my time. I actually was in Asilomar as a very young fellow. Somebody, Hans Zachau, took me with him I was a nobody, so to speak, and so I remember some of these discussions. At that time, in 1975, there were guidelines proposed, later enforced by the National Institute of Health. In Europe, nobody knew about this at the time. It had all developed in California, even though one of the discoverers of the prerequisites for this, Werner Aber, a professor in Geneva at the time, now in Basel and still alive, discovered restriction enzymes. I mentioned that yesterday as another mechanism of bacterial immunity, restriction modification. So that was 40 years ago, and in fact, it was very bad even in the US. The mayor of, what was his name again, in Cambridge, Cambridge, Massachusetts, wanted, in fact, close down the entire city and forbid experiments with recombinant DNA. And there was a popular vote at the time, and what was his name? Zanetti? Oh no, an Italian name, but I come to it while I talk about something else. So at that time it was very critical, and then but the guidelines were relaxed rapidly because it turned out that these cloned pieces were much less dangerous than the entire virus, and all the arguments since then, of course, as you all know, you could clone SV40 in a high school experiment because it's well known that it doesn't create any problem. But at the time, these guidelines were developed, and it was pretty bad in Germany. At least I was also involved in cloning. We looked like astronauts when we cloned bacterial genes because we had to really dress up, and it was very complicated at the time. But it's all forgotten. Anyway, but the people like David Baltimore, a Nobel Prize winner on reverse transcriptase, and others, Paul Berg, remembered what they did 40 years ago, and so convened this conference in Napa Valley in California. And out of this conference came a committee, and they made me a member of a small committee, I think we were 12 people, to prepare for a conference in Washington, which took place in early December, so-called an international summit on human genome editing, gene editing, and which took in fact place. It took quite a while to organize it and find the proper people in order to discuss these issues. So about 600 people attended this from all over the world and discussed the consequences of CRISPR-Cas just on the human question, not on plants and not on mice or whatever, which is another matter, but on the human system, based basically on the experience that the Chinese had already done it. Turned out, by the way, the Chinese, you may think, do everything, but it's not true. The legislation in China is very strict, actually. If you now investigate, and we did, the legislation around human embryo work with human embryos, it is in many countries, including China, highly restricted. In fact, 40 countries in the world have restrictions on the use of human cells and human embryos, with any independent of CRISPR-Cas. Okay, so the conference took place. This is the lecture hall of the Academy. Some of you, Glauco is a member. Some of us have known it because we are members, but and at this, during the conference and as a result of the conference, this small group of 12 people prepared a paper on human gene editing, and the conclusion of the paper, as you will see in a minute, also was to transfer the news, the ideas about this problem to other countries so that they would introduce legislation wherever necessary against using in favor of doing these kind of experiments. And so I will just go through the recommendations of this particular conference, which took place under actually was not just the National Academy of Science, it was the Chinese Academy of Science and the Royal Society, those three. The other countries like Germany, for example, France, had already also, even before that date in December, concluded that something had to be done. I will come to that and we can discuss it. I'm sure I cannot really discuss everything surrounding this issue because it really has become quite hot. So I will go through these conclusions, and they are like this. The first chapter was on basic and preclinical research, and here a lot of arguments were brought up in favor of doing experiments with human embryos, as long as the modified cells were not used to establish a pregnancy. So what would you want to do? And what are the prerequisites if you want to work on human embryos, which are not used later on to establish a pregnancy? And you can see that here. You want to do that because you want to understand the biology of human embryos. If you think this is doubtful or something, please keep it in mind and let's discuss it. I mean, there are people who think the biology of human embryos is already known because it's very similar to the mouse. I come to that. It's not true, actually. And then the benefits and risks of clinical uses, and of course the technology of doing CRISPR-Cas and gene editing in the human system. Now the arguments. Why would you want to understand the development of the human early embryo? Now there's some arguments which I brought up here. Janet Rossett is at the Children's Hospital at the University of Toronto, who are very far in this business. So you want to know the early beginnings of human development, improvement of in vitro fertilization, which is still not very good, but it works, of course, as you all know, prevention of early pregnancy losses, and understand the origins of pluripotency. And as you all know, this is not a trivial question if we think about producing human embryonic stem cells out of human embryos. An experiment actually which was done first in 1998, so it's almost 20 years old, which already then created a lot of problems. The problems, well, so one argument is, of course, that people say you don't have to work with human early embryos because you can do this all in the mouse or in experimental animals. The answer is not so easy. There's an interesting paper in, I think it was in Development last year, in August or so, where it really shows that if you do a deep sequencing of RNA, we heard about this earlier today, if you do deep sequencing of RNA, you realize that there is an overlap between the human embryo proper and the mouse ICM. Actually, they look quite different. It's very interesting that a human embryo and a mouse embryo with this inner cell mass and so on. I think we saw some pictures earlier are looking quite different, but this is more instructive is this RNA analysis. You can see there is an overlap here. You know, some of these genes, SOX2 are the Yamanaka genes, which you need to make a fibroblast into induced pluripotent stem cells. There are others here. Fibroblast growth factor 4 is one of them, in any case, you can read it here. These are the two important ones, but there are many others which are unique to the mouse or unique to the human embryo proper. So there is an argument in favor of working with human embryos. Now, in fact, in Britain, at the Crick Institute, that new institute at the, what's it called, St. Pancras Station, there's this gigantic new Crick Institute, wonderful place. They have applied as a human embryonic fertilization agency in Britain for exactly these type of experiments, and they got approved recently, and they can, in Britain, you can work with human embryos for 2 weeks. So you can even work with embryos proper, and not just with these pro-nuclear embryos, but with real embryos. And as long as you don't establish a pregnancy. For 2 weeks, it's permitted in Britain, as long as you get permission from the Human Fertility Agency. And they got the permission. Clinical use. So, as you can see here, there's a lot of potential, if you think about it, of using somatic cells, not germ cells, not embryos, but human somatic cells we are now talking about. We are now talking about somatic cells, and changing them in such a way as to treat disease or prevent disease of all kinds. So the best example, I mean, the somatic cells are really an example which was discussed in a very positive way, because these genomes or these cells are not transferred to the next generation, and therefore there is no difficulty in trying to recall genes which you think are not working properly. And we always think of the next generation. As I alluded to in the last section, if you start making blue eyes, then which would be easy, then everybody suddenly would have blue eyes. Now the question is that useful? We come to that, by the way, in a second. So what could you do with somatic cells in the clinic? And it's already something which is already in the clinic. Is this here? Is, as you know, there is a small portion, 0.01% of all people are HIV resistant. And they are HIV resistant because they have a mutation in a gene, in two genes, CCR5 and CXCR4, which prevents the HIV virus from entering into T cells. You know that HIV is a T cell disease, so the T cells in the body are affected, killed, and with all the consequences. And in fact, some of the consequences only appear 10 years later if you don't treat properly. And then the virus is coming back to the patients. And then actually this receptor here, CXCR4, is the one which is then the important receptor after the disease comes back, if it's not treated properly with antiviral, antiretrovirals, and so on. So that experiment can be done. You can take T cells of any given patients and CRISPR-Cas them with this procedure, homologous genetic recombination, introduce a mutated CCR5 or CXCR4 gene, and then gives the cells back to the patients, and then they have an immune system which has T cells with that particular mutation, makes them resistant to HIV. Now, I think at the last week or so, there were about 40 patients at the Mass General Hospital which have been treated like this. So, I mean, T cells are somatic cells, they are not in the germline, and the patients are alive and well. And of course, the experiments, high-risk patients, so the experiment has to wait whether it works or not. And there will be more and more patients, people will be treated this way, and then one has to wait whether they ever get HIV or not, as compared to a control group. This is not known yet because it was just done in the last couple of months. So I have no result. But just to tell you that, yes, there are clinical, these kind of things are in the clinic, and people have already done these experiments. So the conclusion was somatic cells. Experiments can be done as long as it says here that the risks are understood, and there is the major risk is inaccurate editing, which is also mentioned here, I think here. Now, you can imagine there's a big discussion about inaccurate editing. You know, you want to have a 3 billion base pair genome, and then you want to introduce a single mutation somewhere. And are there other mutations which you don't know about and which destroy your goals, or which are oncogenic and God knows? And there's very good papers also from the MIT groups who have shown that in this particular case, it's hard to believe, but you can use proper Cas9 mutants which prevent mismatched guide RNAs to create cuts. You know, you remember that you have a guide RNA, maybe 20, 25 nucleotides long, and of course, it can find in a huge genome, it can find mismatched DNA. You can, since you know the sequence, you can find out potentially where these sites are, and that has been done. There's good computer programs, which I don't mention here, but they do exist. And then you can find out the appropriate sites, but you can also modify and mutate Cas9 in such a way as to prevent the binding of the guide RNA in a mismatch situation, 1 or 2 mismatches. And that has been done. Kleinfelter and others have just published that. And you can look that up, Nature, where it was shown by deep sequencing, RNA sequencing and DNA sequencing that in fact there is no worry if you do it properly with a mutated Cas9. And of course the X-ray structure of Cas9 is known, so you know exactly where the single-stranded RNA binds, the guide RNA binds, and so this is developing. So to say that it's a good argument, you need to avoid inaccurate editing, you need to avoid it, but the argument is known and people are working on it, and it can be tested by RNA sequencing and the DNA sequencing. So with that kind of precautions, you can do it. I am not aware of anyone in Europe doing these experiments except in the United Kingdom. The rest of Europe has legislation which prevents that. In Germany, we have a very funny situation, very contradictory. In Germany, an embryonic protection law was created in 1991, this law says that you can use embryos, human embryos. I'm always talking about human embryos for research. This is what it says in paragraph 5. But in paragraph 1, it says you cannot prepare them. You're not permitted to prepare them, so this is the end of the story. Unless you want to establish a pregnancy. Of course, you can do in vitro fertilization, but you cannot produce embryos for research purposes. This is actually true for other countries as well, including France, for example. Okay, so clinical use of somatic cell is permitted under certain precautions, some of which I have discussed. Then comes the more difficult question, the clinical use of the germline, that is now of human embryos. This is hard to read. No, what did I show you here? That's the case of which I had before. So clinical germline modification. There are a number of risks which are mentioned here, probably hard to read. I will go through some of them. If you want to read a paper about this, Eric Lander wrote a paper about this in the New England Journal of Medicine in August with the title Brave New Genome. Brave New Genome. a title which he stole from somebody else, as you know, almost from somebody else. But there was also a paper by him called The Heroes of CRISPR-Cas in the first issue of Cell of this year, where an interesting answer was written. There was a blog saying, I should have brought this, didn't think of it. There was a blog, somebody else wrote, Eric Lander, actually was only born 4 years after Watson and Crick discovered the structure, but actually I discovered it. The Higgs boson, actually I'm not a physicist, but actually I did discover the Higgs boson. And CRISPR-Cas, you know, I talked to God, and I told him to develop CRISPR-Cas, and he did it, by God, he did it. And then there was an answer by another blog who said, I am God, you never talk to me about this. And so this is the kind of discussion is going on with the Broad Institute. And but anyway, the paper, let's get serious. It's why is germline editing a problem, and what would you have to observe before you could ever do it? The risk of inaccurate editing, off-target mutations. I mentioned that before. The difficulty of predicting harmful effects. I mentioned, I think, earlier that you never know if you knock out one gene, whether that hasn't indirect effects on other phenotypes of people. It's hard to predict, and it will be difficult to predict. And of course, you have to consider implications for future generations. How does that affect the evolution of mankind? The genetic alteration would be difficult to remove. And I mentioned earlier today gene drive. By the way, there's just a paper appeared on reverse gene drive. So you can reverse the gene drive. But that you can do in mosquitoes, but not so much, not in people. You know how long the generation time of people is. Of course, you would create social inequities because the experiments would be costly and could only be done by rich people and so on. And then, of course, moral and ethical, philosophical considerations of whether, in principle, you should do anything with the human genome which is non-natural. Actually, we do this all the time. I must confess, although we don't know what we do, but we do it all the time. If in some cultures mothers decide who is going to marry, then this is, of course, a highly genetic process, or this influence, the genetics has nothing to do with love or anything, and that happens, of course, all the time. So the conclusion was, because all this is unknown, the conclusion was that it would be irresponsible to proceed today with any clinical use of germline editing unless and until the relevant safety and efficacy issues have been solved. Are there benefits in the first place? Are there alternatives? And so on. And what it also said, that there should be a broad societal consensus before this has ever been done. Cases of potential benefit are extremely limited. I come to them in a minute, and we can discuss the issues. So it should be revisited, the issue, but at the moment it would be irresponsible to do anything. And so there was not a moratorium. In my country, in Germany, there is a moratorium already on this type of things. But because it was a worldwide conference, so to speak, it was difficult to talk about a moratorium because the legal background is so different in different countries. So it's very difficult to deal with this. In Britain, the government has little to do with these kind of things. In Germany, this would have to be decided by the parliament, and everybody has different cultural and backgrounds. So we said it would be irresponsible to do it now. So what could you do? So, justification, and so now we can discuss what you would like to do. Disease prevention. That sounds as the most responsible thing to do. There are certain diseases which are homozygous, which are like Huntington's disease, where only one copy is enough to create a disease. Other things like sickle cell anemia, which is also here, are recessive. But anyway, I think in the United States, 25% of the black population have sickle cell anemia, 25%. And why do they have sickle cell anemia? Because they come from West Africa. And West Africa, via the Caribbean, they got into the United States. And of course, in West Africa, they had the advantage of becoming relatively resistant to malaria. But in New York, you don't have to be resistant against malaria. That doesn't exist anymore. So the advantage is not there. There's only the disadvantage that you have sickle cell anemia, and it is a disadvantage. After the conference was over, I was just before Christmas, as you saw, the first week of December. So in Georgetown the next day, because I couldn't get a flight back, so I went to an Apple store in Georgetown, which is a suburb of Washington, D.C. You know, these modern temples, transparent buildings, wonderful. I'm not sure whether the Apple store in Munich is not that fancy, but it was really fancy. So I went in and looked at the latest, and I see people here with very tiny. I still have this 1.6 kilogram Mac Pro. And then the guy, black, I have something for you, And then he showed me an Apple Watch. And he showed me that you can have it in every price from $500 to $25,000, with platinum and diamonds and everything. And so I looked at him and asked him, why would I need an Apple Watch? And he looked at me and said, I don't know, but you know, I am black and I have sickle cell anemia. I said, okay, I'm very sorry about this, but what does it have to do with the Apple Watch? It turns out that if you have sickle cell anemia, your blood pressure can jump up and down tremendously from normally we have 120, let's say, to 250 and back. And that is not so funny, actually. And so he has programmed his Apple Watch in such a way that when the blood pressure rises, it makes an alarm. And then he takes his medicine. And so he said, very useful to have an Apple Watch. Well, I said, I probably have other disease, but certainly not sickle cell anemia. I have my genome sequenced, actually. I hope you all have yours sequenced. No, you don't have yet, no. At the Mass General, when you get there as a patient, which I did, they automatically do they do the sequence automatically. This is why I got sequenced. In fact, they think that the other revolution is genome sequencing, that and that's highly related to this, that in 10 years from now, 2 billion genomes will be sequenced. This is 1/4 of the human population, which interestingly is a problem of data at 10 to the 21 bytes, 1,000 to the power of 3, and you can, 1,000 to the power of 7, so you can imagine that, but that's another problem. In any case, I didn't buy an Apple Watch, but not yet, but I think I have to buy one. Everybody tells me I'm so old-fashioned that, do you have one? Who has an Apple Watch here? Ah, there you are. Just one. So next year we'll all have Apple Watches. There are alternatives. You can make PGD because you can, in most cases when things are heterozygous, you get embryos which are not affected. It's only in cases like Huntington where every embryo is affected. And 50% if one partner is healthy and the others are sick, Otherwise, it's only 25% when they're heterozygous. But it's a dominant disease, and therefore, and the funny thing is, Huntington patients always marry Huntington patients. That has to do with the fact that they always meet at the same doctors and at the same circles and so on. You have to, in order to get partnerships, you have to meet somehow, and that is a critical issue in that connection. So you can actually, with PGD, prenatal genetic diagnosis, you can actually select those embryos which are not affected. Now, Huntington is a rare case. Huntington is only 1 in 7 million or so. It's a very rare case, and therefore, maybe there is an argument that there are cases where you cannot have children. That is also an argument. I don't dare to say it, but I think in these such rare circumstances with such severe disease like Huntington, maybe that is what you should consider. But I am not affected, and therefore it's easier said than done. And then came, so there is alternatives in most cases. If you come to these monogenic disorders. And then there is modifying disease risks. I talked about HIV resistance. These 2 genes which are mentioned here, 2 deletions in the CCR5 and in the CXCR4 gene. I mentioned that already. That you could do in principle on a somatic level, on a germline level too, then the entire population would be HIV resistant. It turns out, by the way, that these people, and they do exist, 0.01%, are highly sensible to West Nile virus. Now, nobody in New York or in Trieste I don't know, maybe Trieste is warm enough, but at least in the northern part of this world, there's no West Nile virus. And I think it's in Texas and this more tropical, subtropical parts of the United States, but certainly not in Mexico, but certainly not the northern part. Then there's heart disease, the PSCK9 gene. This is a gene which provides with, which makes you have extremely low cholesterol levels. We all have cholesterol levels of at least 160. We get milligram per deciliter. We get excited when we have 200. Then we start taking beta blockers and inhibitors. Statins. Then we start to eat statins. And there was this woman 6 years ago who had broken her leg in a car accident, which went into the Dallas Heart Center. And they measured the usual blood tests, routine blood tests. They found out that she has an LDL level of only 12, I think. And everybody thought she cannot live with such a low level of LDL. You cannot live, but of course she did live very well. And that then they found that was a mutation in this particular gene, the PSCK9 gene. I think in the meantime, Sanofi has developed a drug against that. Is it on the market? Probably not yet, or soon on the market. So that was 6 years ago, and it takes about 7, 8 years to develop drugs. So you could make the population with low LDL levels. Alzheimer's, there's a good, the APP A673T mutation is a very good mutation which reduces the risk for Alzheimer by a factor of 30, I think. The BRCA genes, of course, as you all know, are predisposing to breast cancer. What was that actress who had her breasts removed because of her mutations? Angelina. She had mutations in both genes. And then you could create resistance to global pandemics like influenza, which would not be too difficult in principle. And then they discussed in the meeting enhancement. So you could, muscularity, eye color. Height is difficult, actually. Height is very difficult because there's no gene known from more than 150 genes known which contribute more than 1% to the height of someone. So height is a very complex phenotype, extremely complex. So if you have, if there are two partners who are very tall, the result may be somebody who is quite tiny, and it may only show up in the second or the third generation, which of course you all know. Eye color would be easy. Learning and memory, high IQ. There's nothing I can say. Maybe you have an idea of how you would improve the IQ. So the conclusion was not to do any experiments with the human germline I mentioned before, and with these arguments in the back. And maybe you have some other intelligent arguments. And then the idea was we concluded the need for an ongoing forum, that since each nation has different legal backgrounds, everything should be studied under the particular jurisdictions of these countries. The academies should take part in this. And I mentioned that the US National Academy and others had participated already in the meeting, but not everybody has attended. So the academies in other countries, here it would be Lincei, I guess, if they are active enough, would have to do this. Germany would be the Leopoldina. In France, in fact, it's done under I just attended an INSERM meeting, and there will be a meeting of the Federation of European Medical Academies, end of April, where the leadership is France at the moment, and they have prepared a paper which arrives this morning of how to deal with that. I haven't read it yet because at 5 o'clock I closed the window and slept again for an hour. So, but I will read it. So the conclusion was not a moratorium, but to discuss it on a very broad basis in the public and with research funders, policymakers, everybody involved, the public, and social scientists, ethicists, bioethicists, and so on. So that is the current state of the art. So there are countries like Britain, where this can be done on with somatic cells in a very limited way. I know of nobody who does it on the level of germline cells, oocytes and eggs. But I think considering what can be done and how easy it is, it would be worthwhile to discuss it, and this is, I guess, why Glauco asked me to come here and present it to you. Thank you. Many thanks.